In the early 1900s, scientists always thought that protein was the genetic material, that it was passed on from generation to generation and not DNA. The reason because protein has more structure. It has primary, secondary, tertiary, and quaternary, whereas DNA, it only has a double helix. But then back then, people didn't know much about DNA. It was so small, it was packed inside the nucleus, and scientists didn't have the technology back then to learn about DNA. Protein are more abundant. It was easy found inside its cell, so that's why scientists thought that protein was the genetic information. Also, protein has 20 amino acids. With 20 amino acids compared to only 4 base in DNA, it was reasonable to think that protein could be the one that coded for all the genetic information. It was not until a few experiments that lead up to the discovery of DNA as the genetic material. It started back in 1928. One of the first and earliest experiments was done by Frederick Griffith. In his experiment, he experimented on bacteria and mouse. At that time, he was studying about the bacterium that caused pneumonia. In his experiment, his first group of cell he took was living S cell. This was a control group. In this group, the bacteria were smooth. That was why it was called the living S cell. S stands for smooth. These bacteria carried the disease pneumonia, and then he injected to the poor little mouse, and then the mouse died. Then he repeated the experiment. This time, he used a different group. He used living R cells. R stands for rough. These bacteria are rough, but they are harmless. They don't have the outside covering that causes pneumonia. Once he injected into the mouse, as expected, because these guys are harmless, the mouse lived. Then for the third part of his experiment, he took the same smooth S cells bacteria and then he heat killed them. So basically he used a Bunsen burner or some sort and then he sterilized by killing all the bacteria. So now those bacteria are dead and then he took those bacteria and he injected into the mouse and the mouse lived. His last part of the experiment, he took the dead bacteria that were harmful and then he also took the living, rough, harmless bacteria and then he mixed them both together. Then he injected into the mouse. What do you expect to happen to the mouse? Well, the mouse died. It turned out that some of the living rough bacteria turned into the living harmful S smooth cell bacteria that causes pneumonia and kill the mouse. So if you were a scientist, what could you conclude from this? The fact that first you injected harmful bacteria that was living, the mouse died, and then you take harmless bacteria and the mouse live. So after you kill the harmful bacteria and you inject into the mouse live, you can expect that, okay, if we were to repeat the experiment, this time with the harmless bacteria plus dead harmful bacteria, you inject into the mouse, it would live, but it didn't turn out that way. It turns out the mouse died. So the conclusion you can make from this is that the fact that some of the harmless bacteria turn or transform into harmful bacteria and then it was injected into the mouse and it kills the mouse. So it kind of have the old term saying like pick your friends wisely, like if you hang out with bad people, chances are you're going to turn and do something bad, and if you hang out with good people, then chances are you might become a good person. So in this case, these rough, harmless bacteria were hanging out with the bad bacteria, and then they kind of took up the bad stuff from them, and then they transform, become like one of them, and then so now they're one of them. So the question is, how and why did they become one of them? How did the harmful bacteria that died pass on the information to the harmless bacteria? Well, this question is going to be answered in the next experiment, which is done by Avery. So Griffith discovered that bacteria can transform. As for the question of how, we have to wait for Avery experiment to find out.